Now, this is something that we know as DNA, and sometimes we can represent this DNA by this simple line diagram, this curvy line diagram. So it abstracts those your letter like rungs and the sides of your DNA, which has those sugar and phosphate groups. So you can either draw this one, or just for convenience we can just draw a curvy line to represent this DNA. So when this DNA molecule is wrapped around some proteins called histones, then what we get is chromatin. So these pole kinds of structure that you're seeing here, they are histones. So when your DNA molecules are wrapped around these proteins called histones, then what we get is called chromatin. And basically these histones provide structure to the molecule, and these chromatin threads are very loosely packed and too much dispersed in your nucleus. So if this is your cell and this is its nucleus, then these chromatin threads are just like dispersed, very loosely packed everywhere in the nucleus. Now, when this chromatin is very tightly packed and condensed, then what we get is chromosome. So the only difference between chromatin and chromosome is chromatin is the uh, loosely packed version of your genetic material, and chromosome is your tightly or very highly condensed version of your DNA molecules. In most of the time in your life cycle, we only have this chromatin version of your nucleus, but during the cell division, when cell wants to proliferate, then chromatin condenses and takes up this chromosome variation. But why does cell take up this highly condensed orientation at the at the time of dividing, at the time of cell division? So this uh, tightly packed or highly organized structure of your chromosome helps in segregation of your copied genetic material. So as we know, in a cell, first, in order to divide it, in order to produce more cell, it first has to make a copy of its genetic material, and those two copies will be separated later in the cell division. And it is said that when we have this highly condensed version of uh, genetic material or chromosome, then it is easy to separate them. So it is easy to separate two chromosomes. So if this is your one chromosome and this is your second copy of your chromosome, this is your one chromosome, this is second chromosome, which is copied from chromosome 1. Now it is easy to separate them and make two new chromosomes when they are very highly condensed. But when we have chromatin only, which are very dispersed in the nucleus, then to separate them it is difficult, and we some errors can occur while separating loosely packed or dispersed threads. For an analogy, I can tell you that you can assume two woolen balls. So if they are very dispersed, let's say we have a red wool, red threads and blue threads, they are very much dispersed and mixed together. In one scenario you have like this, and in another scenario we have very tightly wounded balls of red woolen ball, well, red wool and blue wool ball. Then it is easy to separate in that scenario rather than in the messy kind of structure of your threads. So that's why chromosome orientation or chromosome version of your genetic material is required at the time of segregation of your genetic material. After they are separated, they again will get back to their chromatin form in their life cycle, in a life cycle of a cell. Now we'll talk about chromatid. So now we'll talk about chromatid. So this is your chromosome, When the genetic material is duplicated, this is how it looks like. So this happens when your cell get matured and its genetic material is copied and it is ready to split into two new cells. So this in this we you can see this is one copy, this dark red, and this is light red is second copy. So this first copy is called as one chromatid and second copy is called second chromatid. So basically they are called as sister chromatids. Overall, this molecule is will be called as one chromosome, single chromosome. So till the time these two copies, which are called chromatids, are tied together, they are not separated. This overall the overall molecule is called as single chromosome, and in this where we don't have any where genetic material not even copied, that is also a single chromosome, and sometimes people say that this is a single chromosome with single chromatid. With single chromatid. That is just a convention. Chromatid is a bit confusing, but these are the conventions. It is called as chromosome with single chromatid, and it is also a single chromosome with two sister chromatids. When these chromatids are separated, then they are called as individual chromosome. So this is a new chromosome, single chromosome, and it is also a different chromosome. So one chromosome turned into two chromosomes. So here we have one chromosome and here we have two chromosomes, and this one is a chromosome having single chromatid and this one is also a chromosome having a single chromatid. So till the time they are not separated, they are a part of only one chromosome and they are called chromatids. Later when they are separated, they will become into they will be called as two new chromosomes with each having their single chromatid. So this was all about chromosome, chromatin, and chromatid, and I hope you got the difference between these. Chromatin is your lightly packed version of your genetic material. When it is highly condensed, then we get chromosome, and these copies of your genetic material of your chromosome, they are called as sister chromatids till the time they are not separated. After they are separated, they are again become two new individual chromosomes.